Hi and welcome to my OCR A-level biology revision session with me, Christine. So today's lesson I want to look at the loop of Henle as part of your kidney topic. So let's just remind ourselves about the structure of the kidney nephron then. So we have our glomerulus and our Bowman's capsule. We have our proximal convoluted tubule. We're then going to go down our loop of Henle, which is what we're going to focus on today. Then our distal convoluted tubule, and then the collecting duct leads down into the pelvis. And the loop of Henle and the collecting duct are the only parts which are located in the medulla in the kidney. So the role of the loop of Henle is that it is going to enable the mammals to produce urine which is more concentrated, and the loop of Henle can differ in sizes depending on the animal and depending on the environment for which they live. If they require to retain more water inside the body because they live in areas where water is in short supply, then you would find an animal that would have a longer loop of Henle. So let's just look at this loop of Henle then and identify some key features. So the loop of Henle has got these different areas which have different permeabilities to water, and that's really important in its role to play to ensure that we maintain the water so that we can keep an osmotic potential that our cells can function with. So if we go down our descending limb, what we can see then is that this one is permeable to water. Our ascending limb is impermeable to water. What does that mean if it's permeable to water and impermeable to water? Well, what it actually means is that the permeability is to do with the fact that the membrane has a channel which allows the water to move across. Remember, your phospholipid bilayer is going to have the hydrophobic region which will repel water. So therefore, to ensure that we increase the permeability, we need there to be aquaporins, which are water channels that allow the water to be able to diffuse down its water potential gradient by osmosis. So you can see in my diagram that I have my blue channels and I have red channels. Well, the fact that we have the descending limb which is permeable to water, that tells me that the blue channels are aquaporins which allow water to move across the membrane, whereas we have red channels on my ascending limb, so they need to be involved in the transport of a substance that is not water. And what we now know is that the loop of Henle actually acts as a countercurrent multiplier. And what that means is as the nephric filtrate makes its way down the loop of Henle, down from the proximal convoluted tubule, at the end of the proximal convoluted tubule, it is at an isotonic point with the blood. As the nephric filtrate makes its way down the descending limb, what happens is it becomes hypertonic. As you get to that hairpin bit at the bottom, it's losing water as it goes down the descending limb. Therefore, it is becoming more concentrated inside the kidney nephron. As it then makes its way up the ascending limb, what's going to happen is the nephric filtrate is actually going to become hypotonic. Now, to become hypotonic, something has to happen to the solute concentration that's inside the nephric filtrate, and that's where the ascending limb comes in. So we're going to start on the ascending limb, and to know how this all works, what we need to understand is that as the filtrate is making its way up the ascending limb, sodium ions are going to be actively pumped by active transport. They're going against their concentration gradient, so they're going to be actively pumped into the medulla. And what that's going to do is that's going to maintain a really high salt concentration inside the medulla, in the interstitial fluid. Well, the medulla interstitial fluid is the tissue fluid surrounding the kidney nephron. So this is the medulla section, and therefore what we're doing is pumping the sodium into the medulla, so we have a high salt concentration. Well, because we have a high salt concentration, and because we remember that the descending limb is permeable to water, that's going to mean that the water will move out of the filtrate into the interstitial fluid, and it's going to move by osmosis, and it's going to move down its water potential gradient. And that's what we mean by a countercurrent multiplier. By pumping the sodium ions out, what we're creating is a high salt concentration in the medulla. Therefore, the water will leave from the descending limb, and it will make its way into the medulla down that water potential gradient. I hope you've liked this video. If you have, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, please do check out my revision platform, www.aiqchat.com.